Welcome to Unit 7, Lecture 1 on Patterns of Inheritance. Learning targets. Understand the history of genetics. Understand the genetic laws of segregation and dominance. Understand ways to analyze genetics. Identify pedigrees and Punnett squares. Genetics, genetics has been around since the beginning of time. However, the beginning of life. However, we haven't understood genetics. Okay? We've been trying to understand how traits get passed on for thousands of years. For the most part, for a lot of human history, we've gone basically just on phenotype, which is look, physical features. All right, and we've said people are part of the same family just based on some very primitive physical features. However, we now understand that that's not the case. So Gregor Mendel finally started to get it in the 1860s, and he was a monk who was working on peas in a garden, and he identified that there are inheritable factors that get passed from generation to generation. Now, Mendel just called them factors, didn't quite know what they were, because at this point you actually know more than Mendel did when he was discovering this kind of patterns or these patterns. Because what you know about now that Mendel didn't was DNA. Mendel didn't have any concept of DNA or chromosomes or mitosis and meiosis. And so he just figured out that there are inheritable patterns that get passed on from generation to generation. And now we know those factors are DNA and they're on chromosomes. Okay, so over here you have a picture of two chromosomes, all right? And then these what are called loci, which just is a location on the chromosome. That's all it means. It just means location. And if you remember talking about Punnett squares in middle school, you always had two letters. Well, you have two letters because remember you have two chromosomes, one from the sperm that made you and one from the egg that made you. And those two chromosomes come together to give you your genotype, okay? And your genotype has been, historically we took a look at it as two letters, okay? Usually letters of the same, or the same letter. Sometimes it's homozygous, which means two of the same, and this one happens to be homozygous dominant, okay? Homozygous for the recessive, which we usually use a little letter, lowercase letter for, and then heterozygous means one of each. Okay, so you can see here on this picture we have loci for capital P, which are both dominant, lowercase a, both recessive, and then capital B, okay, which is the dominant version, or lowercase b, which is the recessive version. Okay? So Mendel did some experiments with pea plants, and he was looking at a result of 7 different crosses, okay, in pea plants. So he was looking at flower color, he was looking at position, seed color, seed shape, so on and so forth. If you notice, most of the peas we eat are actually green, which is the recessive trait. It's actually the recessive color. Usually peas are supposed to be yellow. Who knew? Apparently Mendel did. Okay, and then wrinkled is the recessive. So sometimes if you have peas for dinner, you might see some of them are round and some of them are wrinkled. Okay, and so what he did is he took pea plants of different characteristics and bred them together to see what would happen over the course of generations. So here in this example of purple flowers and white flowers, you have purple flowers, which are dominant, okay? So we could say they were capital P, all right? With white flowers, which if you see here is recessive, so that would be lowercase p, lowercase p. Of course, p is not a very good letter to try and show you capital and lowercase. Oh well. Okay, they made it together to make all heterozygous purple flowers in the first generation. Okay, all right. Then what Mendel did is he allowed the peas to this generation to then have kids themselves, have offspring. And so he ended up with 75% of them, roughly, had purple flowers, while 25% of them had had white flowers. And so, if we did the Punnett square, which you may or may not remember from middle school, all right, so we're doing the Punnett square for the F1 generation to become the F2 generation. They're both hybrids. Okay, so we get big P, big P, big P, little p, big P, little p, little p, little p. So all of the flowers that had one dominant allele, one capital P, came out purple. Okay, all right. So 3/4 of the boxes, okay, which equals about 75%, all right, came out purple, whereas one box, 1/4, came out white, okay? And so what Mendel saw was he saw this inheritance pattern that allowed us to start actually seeing how traits, phenotypes, which is the physical trait, okay, so purple would be a phenotype, gave us came from different genotypes. All right, and he was able to show us that. So the second law was Mendel's law of dominance, okay, which is caused by, now we understand it as single gene inheritance, which means there's only one gene involved, just one, okay? We're going to get into more complex versions later, but for right now, just one, all right? And so we end up having the dominant and the recessive alleles, okay, which once again just basically means alleles in this image, here, let me pick a different color you can see, okay, is big G, little g. Okay, those are alleles. So that is an allele. Okay, and then we end up with two phenotypes, which just remember is the physical version, dominant or recessive. So for P's, green is dominant. Okay, and white or yellow, all right, is recessive. Okay, and then 3 genotypes, which is basically homozygous dominant, heterozygous, or homozygous recessive. Okay, now in this case, with Mendel's law of dominance, this is the important part here. Okay, the recessive phenotype is only caused by the homozygous recessive genotype. Okay, that's it. All right. Whereas the dominant phenotype is caused by the homozygous dominant and the heterozygous genotypes. So if we were to do this Punnett square, we've got Charmander and Squirtle. Okay. And so just like I did with the P's, although ours are a little bit easier. All right. So the law of segregation, we have separated the alleles. Okay. And then the same thing for Charmander over here. Law of segregation, we separate the alleles, and then you just fill in the Punnett square, right? So the same kind of deal, we end up with 75% of the offspring are going to have a dominant phenotype, 25% of the offspring are going to have the recessive phenotype. Whatever the recessive phenotype might be. I don't know. Okay, but just So that's just what's called a monohybrid cross, because you're just looking at one trait, and it's one little Punnett square. Okay, so when it comes to inheritance, we actually can use Punnett squares, all right, like you saw, all right, to show inheritance patterns or understand inheritance patterns, but we can also use a test cross, okay, or a pedigree. So a test cross is something that will help identify genotypes that follow this regular Mendelian inheritance pattern, single gene, dominant, recessive. And a pedigree will help us actually trace a trait through a family tree. So a pedigree is very similar to a family tree. A test cross is where we end up getting an animal or an organism, a lot of times plants, florists, horticulturists, okay, farmers. All right, we'll use test cross to try and figure out exactly what the genotype is. So in regular Mendelian genetics, a dominant phenotype, so in this case a black dog, has two potential genotypes. That dog could be homozygous dominant, so capital B, or heterozygous. How do we know? Okay, so what we do is we actually cross it with a homozygous recessive individual, so somebody who has the recessive phenotype, and we observe the offspring. If we have any recessive offspring, so in this case any brown dogs, we know, any brown puppies, we know that this dog was heterozygous. However, if all of the offspring are black, okay, we know that they have the dominant phenotype, so we know that it's homozygous dominant. So a test cross allows us to figure out and isolate genotypes depending on what we want. So it all depends on if we're looking for something in particular. So going back to those green peas, if we wanted to isolate green peas because that's the recessive phenotype, all right, we have to just pull out green peas and do nothing with the yellow peas. But if we needed to make a new crop of green peas, we might need to mate the yellow peas together until we got to the green peas, and so on and so forth. Pedigrees, okay, like I said, are just like family trees, except this is a way to actually trace the traits through generations. So not just are we seeing familial relationships, we're also seeing characteristics, okay? So we can use this to see whether a trait is autosomal or sex-linked, which we're going to talk a little bit more in the next lecture. And we can tell so who's affected or not, if it's dominant, recessive. We can see a lot of things. Doctors actually still use pedigrees, especially when tracing cancer through generations, to see if it's a genetic version of a cancer or if it's a environmental mutagen type, from something in a lifestyle. Okay, and what pedigrees do is if we end up having a circle, that means it's a female. Okay, all right, actually let me change colors here. All right, a circle is a female, a square is a male, okay, and then a filled in square means that that male has it, all right, and a filled in circle means that female has it, okay. And another thing that you'll see in pedigree charts, especially for sex-linked, is you'll see that it's half filled in. Okay, and that half filled in means that that person is a carrier, but we'll talk a little bit more about that in the next lecture. All right, I know this was kind of long. There's a lot of good stuff in here though. Okay, I will talk to you later. Have a great day.